what did we look at last time? What were we looking at? What was the purpose of what we were looking at? The protein function.
Helping things get in and out of it.
Yeah, helping things to get in and out of the cell, right? And so things don't like to diffuse through the membrane. Okay, they typically don't, except for what?
Sodium.
Okay, water can get through. Okay, it is because it's in such high concentration that it can get through. All right, what is the other thing that could diffuse right through the membrane?
Fats.
It has to. It isn't fat, but it has fatty type of. It's very hydrophobic. So let's say drugs. A lot of them can actually diffuse. Some hormones that are natural can diffuse right through the membrane because the barrier is a hydrophobic barrier. So if anything hydrophobic can probably get through. But most things are soluble in water because we are made up of water, and so most things get to that hydrophobic barrier and stop. All right? So if it stops, then we have to have a mechanism to get it through. And so we were looking at channels and pumps. What's the difference between a channel and a pump?
Energy versus energy.
Yeah, energy versus not energy. So which one's energetic? I have to invest energy? A pump. Okay? And so a lot of the pumps have a name in them that it is something ATPase. What does ACE mean?
Enzyme.
An enzyme, or to break it down most of the time. I'm breaking something, and what am I breaking? ATP. And so now, when I give you something like an example of the sodium-potassium ATPase, that kind of tells you what's going on, right? That sodium and potassium are moving, but that doesn't tell you everything, does it? Okay? What does it tell you? What do you know about the sodium and potassium? What are some details? Do they move exactly the same way? In the same direction? No. Okay, so what goes outside the cell? From inside to outside the cell?
Sodium goes out.
The sodium goes out,
3 to 2, right?
and the potassium goes in, and there's a ratio, right? How many go out? How many sodiums? 3. How many potassiums come in? 2. So we create both a chemical gradient and an electrical gradient. We call it a chemical electrical gradient, right? Now, what does that create for us? What we've done really is we've converted the bond energy, basically the enthalpy of the ATP, okay, which is highly energetic if I break it, and we've converted it into a gradient because there's going to be a differential. That gradient is an energy store also, all right? And so in this, it was part of a pump that was classified as a P-type pump, and the P comes from what as part of this mechanism? Do you remember?
Say that again. Sorry.
What was the P portion of this P-type pump? Why do we call it a P pump? Not protons. It gets phosphorylated. In particular, an aspartate residue gets phosphorylated, and we can identify the number on each one of the versions of whatever pump we're looking at. But essentially, an aspartate residue is phosphorylated as part of the mechanism. Now, the workings of that pump. How did it work? How did it move things from one to the other? Did it create a hole and everything just flow? No, it didn't. It had an orientation from one direction that had specific affinities in one way, and then it opened up to the other, and then its affinities changed. Okay? So that conformational change opens from one side to the other to allow movement, and as that conformational change occurs, in one sense, we have a very high affinity for one of the ions in one of the conformations and a low affinity for the other, and that reverses when we move that conformational change. Okay? Calcium is also an important ion within the cell, as well as sodium and potassium. All right? And so calcium is a very controlled ion, the cytosolic concentration is about 100 nanomolar, and the extracellular concentration is about 1.5 millimolar. That is a huge difference. Okay, it's 4 log units difference, or 10,000 times different from one side to the other. The low level of calcium on the inside of the cell, okay, allows the release of calcium to be a messenger. Okay, now we trap calcium outside the cell and in vesicles, okay, and we keep it sequestered. The one that we most often talk about are the ER and the SR, where calcium is placed inside, but it's not roaming around the cytosol. Okay, and so ER, SR. So we tend to control where the calcium is. Now, to control where the calcium is, that means we need to pump it from the cytosol into these vesicles or outside the cell. During muscle contraction, which is a lot of the tissue mass in our bodies, calcium has to rise. Okay, and contractions will occur until the calcium concentration goes back down. And so, to lower that calcium concentration, we use two types of pumps. One, the calcium ATPase, which is about 80% of the SR's protein, and the sodium calcium exchanger. Here is the calcium ATPase, also called SARC in literature. And so in this process, it is very similar to the sodium potassium ATPase. All right, one other thing about the P-type proteins or pumps, there are 3 domains: the A domain, the P domain, and the N domain, and you can see them outlined here. The N domain will be where ATP is bound. The P domain is where the phosphorylation event occurs, and the A domain is part of where we bind the actual entities that we're transferring. And so if we start at E1, the first thing that happens is calcium and ATP bind, and the ATP bound, the calcium bound is it's faced towards the cytosol, okay, and we want to pump it into the SR. This is mainly where the orientation happens. The ATP will then be broken down into ADP and PI. In that process, one of the aspartate residues in the P domain gets phosphorylated. That phosphorylated state will cause the conformational change, meaning that it was once opened to the cytosol, it is now open to the in the interior of the SR. And so they call it eversion. In that conformation, calcium binding really is very low, the affinity, so it floats into the SR. Once that is released, there is a small conformational change in which the phosphate is allowed to hydrolyze or be broken off, which will cause it to evert back to its original state. Okay, and so it has this cycle that goes around. The ability for the SR, or for this calcium ATPase to work very efficiently is based upon this, the affinity of these sites right here. Okay, so how good does this bind under a certain concentration, and then the overall rate of inversion, how fast it can convert, will overall be the limiting factor. We have a pretty good crystal structure of this. We have the domains. We can actually see where the calciums bind. We know where the aspartate residue, in this case is the 351 version in humans, and the N domain where the ATP binding site is. Okay, and in this, the stoichiometry here is 2 calciums for every ATP bound. The next is the sodium calcium exchanger. The sodium calcium exchanger is a pump that is going to be our first example of a secondary transport. Okay, it can only work once we have the established sodium potassium or the sodium gradient. Okay, and I'm going to use that energy. To do this, what's going to happen is 3 sodiums will flow in, 1 calcium will flow out, okay, and this is moving calcium outside the cell. We will create a gradient in this case because we are moving differential numbers of positive charges, okay, and so this actually has the capacity to actually move. This is actually a fairly quick enzyme in which it can move a lot of calciums. Now, to lower the calcium concentration inside the cell, we got to do it quickly. The sodium calcium exchanger, okay, and you need to understand this, has a low affinity for calcium, but a higher capacity. The calcium ATPase has a higher affinity for calcium, but a lower capacity. So what does that mean? All right. The sodium and calcium exchanger doesn't bind to calcium really, really well. Okay? It takes a high concentration of calcium to bind to it, relatively. Okay, and these are all relative terms. But it moves quickly. And so it will move more calcium, all right, because it just can do it faster. But it will lower the calcium concentrations in the cell to about micromolar levels. Okay, that's not where I need it to be. And so the initial big push is to push it all out and get it down to micromolar. That's still high enough concentration for signals to be processed. Then calcium ATPase has been working all along. It can bind to very low concentrations of calcium, but it works slower. And so at the initial, both of them are working, and both of them are pumping. The sodium calcium exchanger moves a lot of calcium out, but then it gets to where it's not effective. It's not binding to calcium very much. Then the calcium ATPase is continuing to work, and then it becomes the main workhorse, and it brings it back into the nanomolar range and gets it to where the resting concentration is. So they both work initially when calcium's there. The sodium calcium exchanger moves more calcium but can't get it all the way down. And then the calcium ATPase, although it had been working, becomes a workhorse to get it in its final concentration. Another transporter that is seen is called an ABC transporter. It works fairly similarly in terms of its transport, but in this case it will not be phosphorylated and it will not be able to be inhibited by vanadate, which the P transporters are. All right. So what happens is the substrate, whatever it's going to bind, okay, so here, can either bind directly or a lot of times they're carried. Whatever is going to be transported is carried. So it might bind to a carrier, come and bind to the transporter itself. All right. What happens is the transporter will deposit it inside the transporter. Okay. So now it's on the inside. It will close one side and it will break ATP on the other, causing conformational changes where it opens to the other side. And in this case, I am moving from low concentration to high concentration. So it's investing energy, it's going against the gradient. It breaks ATP to cause these conformational changes. ADP, PI fall out, ATP fall back in, and it will evert to the original state. There are several examples. They did not give a detailed in the book. They told you that in bacteria there was one that transported molybdenum, one that transported a sugar. There are examples of these, but I'm not going to show a particular one. I just want you to understand the ABC transporter class. Here is the second secondary active transport example that we have, and that's the lactose permease. Okay, the lactose permease is going to use a gradient, but this gradient is going to be protons. Okay, and so lactose is here and a very large concentration of protons. Here, low concentration of lactose. Here, high concentration of lactose. Here, high concentration of protons. Here, low concentration of protons. And so I will bind 1 and 1, a lactose and a proton, and I will release them. And so I've got more lactose over here and more protons over here. What kind of transport is this? Classify it. How are they moving? Is it uniport, symport, antiport? Symport. Okay. And once this is released, I evert back and I can pick up again. The energy source is the proton gradient, and what's being moved against the gradient is lactose. Okay. What is interesting about this is that cyanide will inhibit this transport. Okay, have you ever heard about cyanide? Is that something that's good for us? No. If you watch all the crime shows, you can identify cyanide because it smells like what? Do you know? Almonds. Yes, it does smell like almonds. And so it is a poison. It's a rat poison. All right. So is the case of cyanide, does the mechanism of cyanide, just because it does inhibit this, is that why we die? Do we die because we can't get lactose into the system, into our bodies? No. Some people don't even want lactose. Okay? Why in the world is cyanide inhibiting this? Cyanide actually inhibits the proton gradient from being formed. Okay? The proton gradient is used to bring in lactose. But more importantly, the proton gradient creates something else. Do you know what a proton gradient will create? Oh, I know you've probably covered this, you may not made the connection. A proton gradient is very important in making one molecule that we need all the time. Not glucose. We're not going to make glucose. We're going to make ATP with a proton gradient. And so really cyanide will, yes, stop lactose from coming in. That's kind of secondary. What it's going to do is prevent ATP from being made. If we can't have ATP, we're all not good. Okay. The binding site has a proton and lactose. Binding site in high affinity opened to the outside of the cell. And then once the inversion happens, again, the affinity, the shape at which it binds is distorted so that it doesn't bind tightly, and then it can be released. Okay. And then once it's released, it allows the conformation to revert back. All right, an additional symporter, okay, and oh, check this out. It actually has the name in it so that you know what's going on, is the sodium iodide symporter. Okay. And in this case, we're going to use the power of the sodium, of the gradient that is created, to allow iodine to come in. And that sodium concentration is created from the sodium-potassium ATPase. So we've learned that one before. And so this sodium gradient is used for a lot of things. And so iodine is able to be transported across a membrane. Do you know which tissue is dependent upon iodine? Highly dependent upon iodine. Maybe, maybe not. Okay? There's only really one tissue type that uses iodine in high concentrations, and that is the thyroid. Okay? The thyroid is part of the endocrine system in which hormones are being released. And so the hormones that are being released are organic compounds that are highly iodinated. So we've got a lot of iodine on them. And so with a lack of iodine, you can cause disease in which your metabolism isn't regulated well. and or you can get what's called a goiter, in which your thyroid will expand. Things about iodine. Iodine is not necessarily in a lot of diets, so we have to supplement it. Does anybody know where we supplement iodine? Salt. Table salt is regulated by the government in what is served, and so it's iodinated, and so iodine is placed into our salt. And so for most Americans, we're good to go on iodine, okay, because we have an overabundance of salt. All right. The other thing about iodine, way back when my parents were in school, they didn't have, or they did have fire drills. They didn't have tornado drills, but they had nuclear fallout drills. And so what did they do? They ducked and covered. So they thought getting under a school desk was going to save them from the bomb, an atomic bomb. Not so much, but okay. In that, a lot of people had bomb shelters, right? And in their bomb shelters, a lot of them had pills to protect them from radiation. Okay, do you know what those pills were? They were simply potassium iodine. Okay, and why in the world? One of the worst fallout products is radioactive iodine. It's a gamma ray emitter. And so we don't get a lot of iodine, but if we do get it in, all the iodine is concentrated in your thyroid. And so to protect us, what you would do is you take an excessive amount of clean iodine. If by chance you were exposed to some radioactive iodine, just by sheer mass, you're not going to absorb the radioactive because you have so much of the clean, and it would protect you, but not really. I wouldn't go walking into a cloud of radioactivity just because you took a potassium iodide pill. But back then, they weren't quite sure, so it was a super protector. Okay. So, transport, the things that we have seen up to this point. We have seen simple diffusion, water, and hydrophobics. We've seen a primary transport, the sodium-potassium ATPase. We've also seen the secondary active transport. All right. We have also seen aquaporin, where we just create a hole and things can move through. Okay. What we are going to see in the next chapter is the ion channel, which we haven't talked about, which is going to allow ions to move, and we haven't talked about transport antibiotics yet. And so we have covered 4 of the 6 types of movements that happen inside the cell. Okay. Oops. Oh, come on. Work. No. It worked before. Maybe it did work. I didn't see it. Okay. Do you see that at the bottom? Muscle alive. What's happening there? It's contracting. What am I seeing right there? Do you know what that is? It could be heart. It is muscle. What we're seeing is a muscle cell. Does that look like the generalized cell? No. Muscle cells are kind of specific. Okay. And so there are some characteristics of a muscle cell that's just a little bit different. Although a lot of the biochemistry that we talk about, the types of processes that occur actually happen in muscle. So most of the things that we describe are happening in this cell. We see some things on the macroscopic scale. We see the Z lines, that is the edges of a muscle cell. Then we see some lines that are called the thin and the thick filaments. Now what happens is the Z lines will come together, okay? They will contract. What doesn't move? What doesn't shrink? That A band, yes, that thick area doesn't contract at all. But those very thin, light areas get smaller on each side. Okay? The contraction of each individual muscle cell is not so great, but as a collective, you can get a large movement. Okay, so we're going to talk about how do we convert electrical energy into motion or movement. The light area is called the thin filament. The dark area is called the thick filament. And do you see that little darker area? That's where the two overlap. Okay, and so they were very creative. Things that were thin, thin. Things that were thick, thick. Ooh. Okay, let's make a cartoon of that. We can see the thick filament and the thin filament are composed of different proteins and different structures. Okay, and so let's focus in a little bit on the thin filament. What is that made up of? They are mainly composed of a single protein that will create a fiber or a long chain of them. The thin filament is made up of actin. Okay, this particular space-filling model is showing you the amino acids that will interact with the thick filament. We can't get this individually unless we really put it into some extreme conditions. Anything that is close to physiological conditions, these things will clump together, kind of like the hemoglobin did and the sickle cell. Makes a long polymer. It's a little bit more controlled than the sickle-celled hemoglobin. And so here's what we see, a bundle of them. They create two bundles that kind of coil around each other. All right, and you can see where we identified those amino acids that are sticking outward. Those are going to interact with a thick filament. Now, we've changed that globular protein into spheres, those blue spheres. The thin filament is not just actin. It also has two components, one tropomyosin and two the troponin complex. The tropomyosin is a coiled alpha helix. So it is one alpha helix and another alpha helix coiled around, and it makes this very long protein, okay, a fibrous protein. Every so often, we have the troponin complex that are made up of three proteins. One, the troponin C, binds calcium. Troponin TN, C, calcium. Got that one? Then we have troponin T. It binds to tropomyosin. Okay, got that one. Then we get troponin I, which binds actin. I don't get that one. Should be TNA, right? But I guess someone didn't want to call it TNA. They call it TNI. I have no idea why they would do that, but just a few names. All right. So T and C bounces to the calcium. T and T binds to the tropomyosin, and T and I binds to the actin. All right. So this is the whole thin filament. For action, the actin is part of it. For the regulation, the tropomyosin and the troponin complex are part of the regulatory event for what happens. Oh, there's the tropomyosin. There's the actin. There is the troponin C in the middle. The troponin, the TNT is the red one, and the white one is the troponin I. Okay, let's look at the thick filament a little bit. The thick filament is made up in bundles, okay, of 6 proteins, which are then bundled together. And so we have 2 heavy chains. They're heavy because they're big. We have 2 chains that are called the essential light chain and the regulatory light chain. So those are the 6 proteins. Okay. In the heavy chain, the tail right here, the tail right here and this area are all coiled alpha helix. It's really, really long. Here, there's a globular head and a globular head that has some function. The tail is that coiled coils, and then there's a hinge site. I'm going to go back real quick. No, not real quick. All right. The hinge site is this piece right here that bends. So here's the bend. It's alpha helix here and it's alpha helix there, the coiled coils. And then there's the hinge side. Okay. There's some names for these, but we don't need to get into that unless you actually read journal articles. We can see a picture of it here, which is kind of messy, and then they've reinterpreted it here. You can see the two heads, the two chains, and the long tail, and you see how it gets bound up into the others. And so we've got pairs of globular heads that are kind of chained together. The globular head is called the S1 domain. It has a couple places that you really need to look at. Now here is where the lever is, and we have the regulatory light chain. Now this is that long alpha helix. Some people have told me that this thing right here has a different color than this. I don't see it, but there's the regulatory chain. I can see the essential light chain. This one is different than that alpha helix. So they sit right there around the hinge part. They help that hinging motion occur. There is an ATP binding site that you can see, and what we did in the other thing was we made that little cave, okay? And then on the cartoon we had a flat spot. That's where the actin binding site, and then we had the globular head like that. All right, the actin binding spot. And so let's take a look at what happens. Here's the thick filament. How do I know it's thick? Because it's thicker than the other one. There's the thin filament. All right, so one's big, one's small. And the thin filament, I'm only going to show actin in this case. I'm going to identify just one of the heads. Okay, and the myosin likes to bind to actin. Okay, the flat piece and the actin love to bind together. Okay, there's myosin, and that's just one part of a whole bunch of molecules. There's the lever domain. There's the globular head, and here's ATP. Now, I show ATP as being together. The two components are PI, which gets broken off in the process, and ADP. So if you put PI and ADP together, you got ATP, the triphosphate. Okay? All right. So let's see what happens. We got ATP, and it wants to bind into the globular head because it's perfectly spaced to have an ATP bound. ATP binds. Woohoo. When it binds, it causes the release of actin. Okay? So ATP binds, actin's released. Once actin is released, that binding pocket is no longer just a binding pocket, but it becomes an enzyme. And it will break the ATP into PI and ADP. So it hydrolyzes only when actin is released. Now, I now have two pieces where I once had one. Once that breaks, the lever domain moves. Okay, here's where the energy is transferred. Once that ATP is broken, the energy is transferred into a motion. The lever moves. Okay. The next thing that happens, PI is released, and when PI is released, it will rebind to actin. Once the actin binds, it will cause two things. The ADP to release, and once the ADP is released, it's under tension. Once the ADP is released, it will relax back into its original movement or its position. But what has happened is it lets go, binds to the ATP, breaks the ATP, moves. Lets the PI go, binds to actin, lets the ADP go, move. And I'm going to continue to move like that, and I'll have little incremental movements, and I will shrink that one cell until it can't shrink anymore, until you get to a point where the strain is too much for that movement to occur. Okay? And it will move. Now, here's the deal. It will not move unless it is signaled to move. And so if you want to contract a muscle, a nerve signal must be sent to the muscle cell. And what has to happen is calcium must be released, and the calcium will cause this to move. Now, what happens is I told you the troponin and the troponin complex and the tropomyosin are part of the regulatory portion. And what happens is the tropomyosin wraps around the actin and blocks the binding pocket between the actin and the myosin. Just physically gets in the way. So myosin and actin love to get together, but the problem is there's a chaperone, tropomyosin, in the middle. Okay? So, in muscle, everything's floating around. You got tropomyosin, you got troponin complex, you have the actin, you have ATP, you have the myosin. Well, the myosin is going to bind to the ATP, it's going to break it down, and it's going to be in that cocked position, ready to move. But its next step is to release the PI and bind to actin. It can't do that because the tropomyosin is in the way. So, in the resting state, the tropomyosin is poised to do the movement, it just can't get to the myosin. Okay? And so it just sits there, ready to bind to myosin. Only will it bind to myosin when the calcium is released. Okay? And so, how in the world does the tropomyosin get moved by the troponin complex? Okay? Sterically, the S1 binding head can't get to the binding pocket in the actin until I move that tropomyosin. Now, what I told you, well, let's go through this real quick. In relaxed muscle, the calcium is about 1 millimolar. 1 micromolar. Okay, most of the calcium is in the SR, and it's placed there by pumps. When I get a signal, the calcium is released, and it goes to about 10 millimolar. That is the binding concentration for the dissociation constant to troponin C. So the concentration just gets to the level to bind to troponin C. Anything below, it's not going to bind very well, and so this causes the movement. Now let's take a look at this. You had 3 pieces in the troponin complex, right? You had the actin, and then you had the one that bound to actin. Which of the 3 bound to actin? The TC, the TT, TNT, or the T and I? The T and I. Okay, so you have the actin and the T and I. All right. What's bound to the T and I is T and C. Okay? T and C binds to calcium. Okay? And what's bound to the T and C is the TNT, which is bound to the troponin. So I've got actin, I've got troponin I, troponin C, troponin T, and then the tropomyosin. This 3-protein complex is going to serve as a hinge. What's going to happen is when calcium binds, it's going to move conformationally, and it's going to pull the troponin T, which will pull the tropomyosin away, which will then open up the binding pocket. Now, the trigger is the troponin C, which is going to have a large movement, but that movement had to be anchored to the actin, and it's anchored through the troponin I. Troponin C looks like another molecule called calmodulin, short name is CAM. It's got two pockets, one in the N domain, one in the C domain. The C domain has an affinity for calcium at about 0.1 micromolar. So at any concentration, the C domain has calcium. That's not the important part. The N domain, though, has a binding affinity for calcium at about 10 micromolar. That is the signal or the trigger. When that N-terminus binds to calcium, it causes a conformational shift. All right? Take a look at that molecule at the top. That's a pretty large shift. You can actually see how that protein will move, and it will move the whole complex out of the way. And so down here in this other animation, what you see is the calcium binding to the pocket. That's the TNC. It will move the entire complex. And then did you see the tropomyosin that was covering all those binding sites was just moved out of the way? And once it's moved out of the way, myosin is ready to bind, and it will contract and continue to move until one of two things. Either I've come to the physical limit of moving that cell, or the tropomyosin lays back down and blocks. Okay? And so we can see the movement. It will move little by little by little, shrinking that one cell. And all of this works in concert with all of the other cells to create a very large movement. Okay? Each one of the movements is really, really tiny. We determined what that movement was by using an experiment just like this. Do you see those little things moving around? Those are little pieces of actin. We made them glow. And what we did was we would put myosin on a cell slide, and we throw in some ATP. And after looking at that, and looking at the distance and measuring it, a very long job for a graduate student, they determined that in humans, each one movement of myosin is about 11 nanometers. Okay? So each movement is 11 nanometers. Now to make the whole cell move, that takes lots and lots of movements, and to combine that all into a full muscle, that's a lot of ATP and a lot of small little movements to get a very large movement. Okay. There's one other particular protein that converts ATP into muscle movement, and that's called kinesin. It contains 4 subunits, 2 big and 2 small. I know that the notes didn't print this out very well. I have no idea why it didn't print out that whole structure. What this does is it moves organelles and vesicles around inside the cell on the microtubules. Remember the microtubules were like little highways, we said, to organize things? What we see is, if we take a look at the globular head here, and this is the coiled coil, if I look at the top down, here's one of the globular heads, here's the other globular head, and you can see the coiled coil that goes down. There is a binding site for ATP, just like we had in myosin, and there's a binding pocket or surface that binds to microtubules, not to actin. Okay? And we're looking at that globular head. This long coil here keeps the globular heads very far away from the end. The end of this creates a unique binding pocket. So these proteins and the end of this create unique binding pockets to bind to things. Really, you think of this like an 18-wheeler. This is the cab up in the front, okay, the power. Here's the hitch where I'm going to put the trailer onto it. All right. Now, if I want to move something big, I don't need it in the way of this, because this is where the motion is going to happen. What's interesting is we've had some crystal structures of the globular heads bound to the microtubules. When ATP binds to myosin, it releases the actin. This is exactly opposite. When ATP binds kinesin, it binds to the microtubule. It's backwards in that sense, but in every other sense, it's the same. I break, I move, I release, and so they're very similar in the steps. It's just when they bind and when they release are different. Okay, here is a animation of what happens. We're going to take an organelle or a vesicle and move it. And check that out. The little kinesin moves down the microtubule. It's kind of like a truck moving things around. Now, that's a really cartoonish animation. Here's a better one.
Includes a network of microtubules created by the lateral association of protofilaments, formed by the polymerization of tubulin dimers. While the plus ends of some microtubules extend toward the plasma membrane, Proteins stabilize the curve of formation of protofilaments from other microtubules, causing their rapid +N depolymerization. Microtubules provide tracks along which membrane-bound vesicles travel to and from the plasma membrane. The directional movement of these cargo vesicles is due to a family of motor proteins linking vesicles and microtubules.
And so, things don't just diffuse inside cells. They are placed very controlled inside that cell and actually moved. The kinesins, actually, if you take a look here, you see this minus and this plus sign? The minus sign is the center, where the nucleus is, and the plus sign is the periphery. These proteins will only move in one direction. The way they align to those microtubules will only move in one direction. And so kinesin performs anterograde transport, which is from the center to the periphery. There's a whole other set of proteins called dyneins that will move it from the outside to the in. Okay, and so we have myosin, actin, kinesin, and dynein. I showed you two of them in terms of what they actually do.